Hello, Bio 145 folks. This is our chapter 20, blood vessels and circulation, as well as blood pressure regulation, recorded lecture. You're going to need this chapter 20 PowerPoint, which you can find in test 2 materials. And you may also see me refer to several handouts, which are found in this handout folder on test 2 materials. We'll be looking at the baroreceptor reflex side 1 and side 2, which look like this and this. And again, it's up to you. You can have them downloaded on your computer. You can have them in front of you, but I will be using them. We're also going to be using these two more for me to show you the worksheet, more for you to study from. Blood vessel worksheet side 1 and side 2, which look, like this. Side 1. Oops, that's side 2. This is side 1. And then finally, I also will be looking at, during this video, the chapter 20 fluid exchange worksheet, which looks like this. And let me rotate the view here. Okay. Now this chapter is quite a larger chapter. It once again will have anatomy associated with it in terms of the blood vessels themselves, identifying them. I will be, probably with the blood vessels, I will be making a separate blood vessel video for lab in terms of the identification of them using models. And then I also will briefly today refer to some of the figures that you would see in your textbook that would be fair game for also the either questions on test 2 anatomically or questions with the lab in terms of that. Okay, so, and I'll refer to that when I get there. So initially right off the bat in the chapter, I don't know if that's going to work. Yeah, I'd like to show it like this. So initially, right off in the chapter, there are several pages worth of comparing arteries to capillaries to veins, and you can actually see them here in terms of figure 20.1. Now the worksheet, I'm going to kind of quickly go through some of the characteristics of these, but this worksheet, study worksheet, which you do not have to turn in, is very good to kind of use as a front and back to sort of summarize what I'll talk about initially in chapter 20 to see if you understand it. So now, typically arteries convey blood from the heart to the capillaries. So arteries carry blood away. Typically it's oxygenated, but as we saw in chapter 19, there were some exceptions to that rule. Pulmonary arteries carry deoxygenated blood away from the heart to the lungs. But typically most arteries carry oxygenated blood. Most veins that transport blood from the capillaries back to the heart would contain deoxygenated blood with the exception of pulmonary veins. The capillaries are obviously the fluid exchangers microscopically that are exchanging the substances coming from arteries, fresh oxygenated blood. And then the waste and carbon dioxide will leave the capillary bed. We call it the arterial end here and the venule end here. And then we'll end up leaving the capillary bed and entering the venous system and going back towards the heart. And let me see if I can point out some things here. Now with arteries, because they're carrying blood away from the heart and it's under high pressure, they're going to have a middle layer that's much thicker called the tunica media. And you can see the difference between the veins and the arteries here. Now arteries have to withstand pressure, so they're called pressure reservoirs. And that's why they contain that big tunica media to be able to stand the high pressure. Whereas veins don't withstand as much pressure. They're more involved with holding volume. And so they're called volume reservoirs. And so they have a thinner tunica media. because we want them to stretch a little bit more. It's a V. It's hard to write. Sorry. So writing on my screen sometimes can be a challenge. Reservoir. All right. So because they have to hold a lot of volume, and typically when you think about veins as being volume reservoirs, most of your blood when you're standing upright is in your lower extremities, and these veins obviously then have to expand a little bit. In the tunica media, we wouldn't want it to be too thick because we wouldn't be a good volume reservoirs in terms of that. Now, arteries and veins have 3 layers. The middle layer is called the tunica media. The innermost, so this is the middle layer. The innermost layer is called the tunica intima, and this is where blood would actually touch, okay? So if you look over here in this next slide, you can actually see the vein with a thin tunica media and the artery with a thick tunica media, and then you can see the blood in the lumen here, which would be basically the lumen is what has created the space, and it's lined with tunica intima. And so you can see veins have larger lumens because they're don't have as much tunica media, whereas arteries have smaller lumens because they have more tunica media. Okay. Veins and arteries also have an outermost layer called the tunica externa. So this goes from outermost to innermost. So if you were to take a knife and you were to cut through an artery or cut through a vein, you'd first cut through the tunica externa, then you'd go to the tunica media, then you'd go to the tunica intima. Okay. One other huge difference between arteries and veins, because you can see we list several, is veins have valves. And we'll see why shortly. Arteries do not. All right, so there's some quick differences there. Let's move on. Now, when you compare arteries and veins, and we use the word companion vessels, especially when we identify some of these arteries and veins. Like, for example, companion vessels are arteries and veins that supply the same body region. So let me go and find an example of this. So we have to scroll towards the end here. This is figure 2019. And by the way, when I get out to this, you can see that the blood vessels that are highlighted in yellow would be the ones that you would need to be familiar with for the practical setting. Well, excuse me, there's no practical this summer for the either lab quizzes or questions on exams in terms of that. Now, so you can see, for example, in the arm, you can see the brachial artery, you can see the radial artery and the ulnar artery. But if we go to the next slide, companion lying right next to them, supplying the same region, are ulnar veins, radial veins, and brachial veins. Okay, and so not all blood vessels are companion blood vessels, and we're going to see there's some exceptions to the rule. But like, for example, the kidney has been removed here. There's a renal vein, and there would also be a renal artery. Those are companion blood vessels. But sometimes I just have, a blood vessel that doesn't have a companion, doesn't either. I'm a vein and I don't have a companion artery in that same region. Like, for example, you can see this long vein right here. Right here. Right there. That's called the greater saphenous vein, and it goes all the way down here. The greater saphenous vein doesn't have a companion artery. So there's some that you'll see, the median, the basilic vein and the cephalic vein do not have companion arteries. So you can kind of see that as we look through it. Now, in this figure that we started with in the beginning of chapter 20, though, if you were to look at arteries and veins in the same region, obviously, you could name them the same, brachial, radial, etc. But obviously they're still going to have their different characteristics in terms of where they're sending blood, how they're built, etc., what their job is, right? Okay, so here's another thing that's interesting. So as arteries, the largest artery obviously we have is the aorta. As blood leaves the aorta and as it gets distributed throughout the body, our capillary beds, the arteries get smaller and smaller and smaller and eventually become capillaries. But as blood goes back towards the heart, okay? In veins, veins start out small, the venules and the capillaries, but then they get larger as they go back to the heart. So there's a kind of a difference that you can see in terms of that, okay? That in terms of the other differences that we talked about. Now there are three types of arteries: elastic, muscular, and arterioles, and that's what we're going to look at next. In terms of, as you're looking at these arteries, elastic, muscular, arterioles, and then the capillary portion, the arterial portion of the capillaries, notice that they're, as you're looking at them, they're getting smaller in size, and as we get to the smallest capillaries, they basically just have an endothelial layer that's lined with, the capillaries have a one layer endothelial layer that is thin, a thin wall. It's a small diameter. It's optimal. It's optimized for exchange between blood and the tissue fluid. But as you're leaving the heart, with the aorta and some of our elastic arteries, you can see all three layers are very prevalent. But as I start to get smaller, you can see some of my layers get a little bit thinner. And then you can see even thinner in the arterioles, and then just only one layer in the capillaries. So let's look at the different types of arteries: elastic, muscular, arterial. Now you're not going to have to identify these via microscopically, but it is interesting so that you can actually see the different layers from the tunica intima, where the blood would sit, tunica media, middle part, and then the tunica externa would be the outermost part. And then you can see the lumen in each. So let's look at each of these individually. So there's three basic types of three basic types of arteries. And as we talked about, they branch into smaller vessels extending from the heart. There will be a decrease in lumen diameter as we look at these, a decrease in elastic fibers, and an increase in the relative amount of smooth muscle. And we'll see why physiologically later in the chapter. And so our largest arteries are called elastic or conducting arteries. So these guys right here. So the thing is, the largest are the elastics. Their job is to conduct, okay? They conduct blood from the heart to the muscular arteries. Obviously, the largest elastic conducting artery is the aorta. But you also have the pulmonary trunk, common carotid, common iliac arteries. There's more examples, but there's a few terms of that. Now, when you look at their makeup, they have large proportions of elastic fibers allowing stretch and recoil, and that's what we kind of talked about. So you can see, in terms of the elastic fibers throughout the tunica media. So, we want to withstand pressure, but we don't want the artery to stretch way too much. That'd be a problem. That's called an aneurysm, which I'm going to refer to have you watch a video. Now again, we're getting smaller now, going towards the capillary bed. We call these the muscular or distributing arteries. So these guys right here. Now those arteries distribute. Notice their job is not to conduct, but it's distribute. So now you're going to get to distributing blood to specific body regions. The muscle, the smooth muscle, allows for vasoconstriction and vasodilation. So when you look at these microscopically, you can, while you don't see it as much of a thicker tunica media, there would be more kind of smooth muscle in there, so that we can see vasoconstriction and vasodilation a little bit more. Now, there also is a little bit more elastic tissue, and then we can identify it as internal and external. And most of my muscular distributing arteries, when you are looking at blood vessels, are most of your named arteries. And they tell you where like for example, the brachial artery is distributing blood to the brachial kind of region of your arm. The coronary arteries are distributing blood to the heart. So when you think about muscular distributing arteries, you can see that. And then as we get smaller and smaller, my arterioles, these guys, we then see the smallest arterioles only have a thin endothelial layer and a little bit of smooth muscle. And really, these are the guys that help to regulate our blood pressure and blood flow, because we can, if the smooth muscle constricts, we get a little contracts, we get a little vasoconstriction. If the smooth muscle relaxes, we get a little vasodilation. And so when we start to talk about the physiology in the chapter, and you think about how we're regulating blood pressure by regulating the size of blood vessels, it's the arterioles that tend to be probably, when you're thinking about it, the workhorses of of the blood pressure, in terms of that. They play a very significant role in regulating our blood pressure for that. So, in terms of as you're kind of thinking about studying, we've pretty much, and again, when you get to a point of I feel comfortable with material, you could come and try to fill in your study worksheet. Once again, I won't be providing the key till a little bit later on in the unit, so that you're not just filling things in, that you're trying to see what you can remember. And so we pretty much have went through the first 3 items here. And now we're going to quickly go through the rest of the items here, okay, on the other half of the sheet. Now, the clinical features in here, like for example, atherosclerosis, which would be a buildup, unfortunately, in arteries, in which we get this kind of accumulation of fat and cholesterol and plaque, and then it can block blood flow. And this is showing, obviously, one of the treatments, especially if it was a coronary artery in the heart, where we would actually put a device called a stent in place. And I think if you watched one of the videos from chapter 19, and actually let me go back and show you that folder again. So test tube materials. Any videos that I refer to for you to watch right here, videos and animations. So there is one on heart attack due to atherosclerosis, and I also mentioned that in chapter 19. Check that out if you haven't done that yet, okay? And you can see that one of the, and that video will also talk about CABG, C-A-B-G, which is coronary arterial bypass. Let me write that up here. Oops. CABG. And so the video will talk about that, about how you're actually bypassing coronary artery bypass graft or bypass surgery. That's basically what this is. So if somebody had a blood clot or a blockage in a coronary artery due to atherosclerosis or had a blood clot, in some cases, a stent would be hopefully the least minimally invasive, where we'd thread a special device into a femoral artery and then make its way to the coronary artery using radiography, and then placing this kind of mesh-like device to sort of open up the artery. Now, not everybody's a candidate for this. If the blockage is too severe, if we can't get the stent in, then we have to do what's called bypass surgery, which you'll be able to see in the video, where I'm literally taking another blood vessel from another part of my body, maybe in the lower limb, and then I'm bypassing the blockage so that I'd have a blood vessel that would bypass it. So as blood's coming in and blood leaves, and typically the bypass goes right to the aorta. So it goes, I'm showing it and bypassing the blockage here. It's kind of like a bridge, but we set it up so that it allows blood to flow to that area from the aorta, because obviously the coronary arteries are what feeds the heart first. So as the left ventricle is pumping blood out of it, and see if I have a picture of this. The video will show it too, but I like to mention it since we're. So, here's a picture of the heart in a bypass surgery. What we're doing basically, and you can't see any of the blood vessels here in the heart, but there would be a coronary artery blood vessel right there, the right coronary artery. And let's say if that was blocked, we literally would, attach another blood vessel that we've had, let me use a different color here, that we've had already surgically removed from the person. And then we would, bypass the area of blockage. And so you'd actually see, and then if you had the anterior interventricular artery here that was blocked, you could do another bypass, if that one was blocked. Now, sometimes you hear people say they had a single bypass, a double bypass, a triple bypass, a quadruple bypass. That means there would be that many more actual bridges. So this would be like a double bypass surgery or a triple bypass or quadruple bypass. So this gives you an idea of what you're looking for here. So watch that video on heart attacks and atherosclerosis. All right, moving on here. Now, capillaries are the smallest blood vessels connecting arterioles to venules, and we started off with that at the beginning of class, right? This is, you can see, beginning of this video, there it is, connecting arterioles, that's an arteriole, to a venule, oops, and they're important in terms of having the ability to exchange substances between the fluid and between the blood and the tissue fluid. There's 3 types. You can see in table 20.2, there's continuous, there's fenestrated, and there is sinusoid. Now, if you notice the pictures, as I'm going from continuous to fenestrated to sinusoid, you're noticing hopefully that the endothelial cells seem to start to, it's kind of like I'm taking an eraser and I'm erasing some of them. And I erased a lot here. It's because of the job and their location. For example, continuous capillaries are most capillaries that we can identify within muscle, skin, thymus, lungs, and central nervous system. The lumen, the lining of these endothelial cells is complete around the lumen. So we don't really have any gaps other than these small little, what we call intercellular clefts that allow exchange. Now, materials that can pass through the blood vessel wall, plasma and its contents, some leukocytes, but not those big plasma proteins or red blood cells, per se, in terms of that. And if they did, we'd have a problem in terms of seeing that. But there are reasons why we need to, have this endothelial cell lining, like in the fenestrated capillary. It looks like someone punched little holes in it, called fenestrations. Now we want more exchange to occur. So in areas like this, the small intestine, the kidneys, most endocrine glands, you'd want a little bit more ability to exchange fluids without being as picky as you are over here. And so large amounts of materials are filtered, released, absorbed, some smaller proteins. And then if you're in areas like this, red bone marrow, liver, spleen, some of our endocrine glands, you need larger gaps for things to get out, like the formed elements, larger plasma proteins. And so you have your sinusoid, where you see the lining of the endothelial cells. Looks like someone, like someone just kind of ripped off pieces of it. The basement membrane is discontinuous, and there is an incomplete endothelial cell lining. But I need that to get more larger substances out. Okay. Once again, as I'm going through these slides quickly, because I can't have the interaction with you and say, are there any questions? You're jotting questions down so that when we do our Zoom meetings, you can ask hopefully questions and or send questions out to me via email. So capillary beds are designed for what we call perfusion, or for kind of fluid exchange, right? And so at any given time, when you're looking at these capillary bed structures, at any given time, only about 1/4 of the capillary beds are open, because there's over 60,000 miles of capillaries. That's a lot. And we really need to be efficient exchangers. And so, and not that you have to remember that, but of those 60,000 miles that you think about, only about 250 to 300 liters of blood are moving through the capillaries at any particular moment. That's about 5% of your blood volume. But we need to be efficient and make fluid exchange rather, easy based on that. Okay, so with these capillary beds, and this is something I'll talk about a little bit later on in this video, there are areas of the body that need more blood perfusion, and there are areas of the body that need less blood perfusion. For example, if you were exercising, your muscles, the capillary beds that would surround your skeletal muscles, we'd want more blood kind of volume, more blood flow, not pressure, but more blood flow coming into that area. There'd be cells and tissues around here. And so what can happen, what's interesting, is we have these little kind of what I call valves. They're actually sphincters. And what comes off, you can see this arteriole, what comes off as a bridge is a meta-arteriole, and then that leads into the true capillary bed. But we have these little, they're kind of, like little mini valves. So if they relax, we get more blood flow. And if they contract, we get less blood flow, right? And so, and that's something that we'd want to happen. And in some cases, we can contract these sphincters so much that blood will just bypass the capillary bed. For example, if my skeletal muscles need more blood during exercise, there's other areas of the body like, where I might not need as much blood, like the digestive system or, blood flow to certain endocrine organs that I might not be using, that type of thing. And so those capillary sphincters would somewhat constrict and or contract and constrict the blood flow. And what ends up happening is the blood goes right through the kind of this thoroughfare channel and doesn't supply the capillary bed. And what we're trying to do is just like in your house, right? You have faucets and sinks and showers and bathtubs everywhere. You are not going to, in your house, turn on every single faucet, right? You're not gonna. And if you did, you wouldn't get pressure. And for most houses, you might not get enough pressure into where you'd want it, and I usually would mention this later, but I used to live in when I was younger, I used to live in a kind of 3-story apartment type of house. And there was first floor, second floor, third floor bathrooms. And if you took a shower on the third floor, you would hope in the bathroom there, you would hope that no one on the second floor or the first floor would be flushing their toilets or turning on a large amount of water, because the shower that you were then standing in would go to a drippy drizzle. So you'd learn to say, hey, don't be flushing the toilet or doing anything else in any of the bathrooms. I'm going to take a shower. I want good water pressure. And so the same thing kind of sort of happens in our body. There are areas where we need more blood flow or more perfusion at the time of, certain times that we would see, and there's areas that we need less. We don't want to just keep the same amount of blood everywhere, because then that would compromise some of the other areas of receiving the nutrients and oxygen and getting rid of the waste that they need to do. Okay, so on the vein side, as we said, veins that connect to these capillary beds are called venules, which you can see here. And then eventually the venules are going to get larger and become small to medium-sized veins and larger veins. Okay, and we talked about that. Now, when you look at the larger veins, most of my medium-sized and larger veins have the valves in them. The small veins, not so much. And we'll talk about why the valves are important as we get into blood pressure and blood flow. And we talked about the fact that veins were blood reservoirs, so I'm not going to go through that again. Remember I said that we have over 60,000 miles of capillary beds. And so what's pretty cool about capillaries and kind of the blood flow is it's kind of, when you think about the greater the total cross-sectional area, the slower the blood flow. And capillaries have the greatest cross-sectional area. So they have more of the ability to have like lots of surface to volume ratio. And you think, well, the blood's not going through there very quickly, but because of the greatest cross-sectional area, more surface volume to ratio, even though they're small, this slow blood flow facilitates the exchange of substances, which we want to happen. The analogy to this, it's kind of an inverse relationship between total cross-sectional area and blood flow velocity. You have more cross-sectional area so that you can exchange more efficiently substances between blood and tissues, but that drops the blood flow velocity. Think about a river. There's an analogy to this. When a river is narrow, if you're a kayaker or something like that this summer, when you go down a narrow river, there's my heart, it's a race. Oops, clear the canvas, there we go. When you go down a narrow river, like this, okay, versus a wide river, this river has more, when we talk about cross-sectional area, we're seeing more cross-sectional area here. This is not one blood vessel. This would be the combination of all the cross-sectional area of capillaries. So, my kayak's going down here. And because there's not a lot of cross-sectional area, the river moves kind of quickly. You're zipping right through. But here, the river's moving a little more slowly because it's wider in terms of that. And as we'll see, this is going to have some, when we start to think about this narrow and wider, we're going to start talking about blood pressure too. Not necessarily in the capillaries, but the blood pressure that's controlled on the arterial end side or smaller artery side. All right, let me just make sure I'm on track here with giving you everything that you need. By the way, the last little bit here, I'm not going to talk about the alternative blood flow pathways. If you are curious, about the information here, you can simply go to figure 20.8 and fill that in. There are 3 alternative blood flow pathways: arterial, venule, and arteriovenous anastomosis. And you can read about those, and they're very well compared in figure 20.8, so you can check that out. All right, so let's do this. So we've basically gone through the dynamics of some basics of anatomy of blood vessels, of anatomy of arteries and veins. And now we're going to get into kind of the physiological portion of this chapter. And so what I like to do, because it is a larger chapter, is to kind of give you an idea of what you're going to be looking at for the physiological side of things, okay? And so we're going to begin kind of the physiology with talking about something called bulk flow, or the dynamics of what kind of happens at capillary beds. Okay. Right. And then from there, we're going to kind of lead ourselves into a little bit of information in regarding the lymphatic system, some clinical stuff. But then the next big kind of physiological portion will be, how do we control blood flow in tissues? Extrinsic, intrinsic factors. It's also good to look at your objectives to see where you're headed, so that you, when you're finished kind of looking at a chapter, you'll know kind of what, not only what we spent the most time on, but what you really need to be, looking for based on that. And so after we get done talking about the control of blood flow in tissues, both the intrinsic and extrinsic control of kind of blood flow, then we're going to be moving into a theme we talked a little bit about in chapter 19, but now we'll spend even more time in chapter 20, called mean arterial pressure, or MAP, mean arterial pressure. And we're going to look at various extrinsic factors that affect mean arterial pressure. We're going to find out what these things mean. We actually we talked about it briefly in chapter 19. Cardiac output, we did, and cardiac output, if we knew someone's heart rate and we knew someone's stroke volume, we could figure out cardiac output. So I could just expand this and rewrite this equation as mean arterial pressure is heart rate times stroke volume times something called total peripheral resistance. We're going to talk about all 3 of these. This is going to be a rather larger topic in chapter 20 for sure. So you'll see me spending a little bit more time on that. And then finally, we're going to kind of in continuation with mean arterial pressure, we're really going to start to narrow down the extrinsic control of it, in terms of how the body controls mean arterial pressure neurally. And we're going to look at the short-term regulation of mean arterial pressure, and that's called the baroreceptor reflex. Okay. Now, once we finish with that, then I'll give us kind of a brief little outline of some of the venous things, and then we'll be finished with chapter 20. But this is a lot of stuff here, and some of the stuff, obviously, in relation to mean arterial pressure, is sort of spilling over from chapter 19. So hopefully you're understanding it, because you're going to have to here in chapter 20. So let me get rid of this, and I'm just going to grab some more water because I've drank all my water and I don't want to lose my voice here. So what you can do, I can't really start and stop the video. You certainly can stop it if you wish, but I can't because of the recording process. So you're going to hear a little gap because I'm just going to run out and grab my bottle of water very quickly. And maybe if you're listening, you don't feel like fast forwarding or whatever, you can jot some questions down, okay? That's kind of what we would do in class. So let me go grab my water, and I'll be right back. All right. So let's see where we're at here. So our first big objective is kind of covering what's called bulk flow at capillaries. So to understand that, first of all, we need capillaries for substances, obviously, to be able to leave the bloodstream and also enter the bloodstream according to their concentration gradient, typically with diffusion forces. So oxygen, hormones, nutrients, carbon dioxide, and waste. You can see, obviously, we'd want oxygen and nutrients to move from the blood into interstitial fluid, and we'd want carbon dioxide and waste to diffuse from the tissue interstitial fluid to the blood. Now, the route diffusion takes depends on the size of the substance, right? And so if you're smaller, you can diffuse right through those endothelial cells that we talked about, or the clefts. If you're larger, you need capillaries that have, when we talked about this, bigger gaps or fenestrations. And so depending on where I'm at will depend on, particle size. And so that's why we have different types of capillaries, depending on different areas of the body where you might need bigger or smaller substance to leave. Now, typically the vesicular transport part of this in endothelial cells, they use a process called penocytosis, and you form those fluid-filled vesicles, and then they're transported to other sites by exocytosis too. Hormones tend to do that. Now, there are typically 3 exchange processes for substances to leave capillary beds, okay? And so depending on where you're at and the size, diffusion is 1, vesicular transport is 2, and then bulk flow, which we're gonna talk about next, is 3. Okay. We don't spend a whole lot of time on vesicular transport. You've already talked about diffusion, but we're going to look at bulk flow because that's kind of interesting in terms of how it works and important to mention. All right. Now, with bulk flow, we're talking about fluids moving down a pressure gradient from high pressure to low pressure. And by the way, just so that you are aware, if you look at it, this is a capillary bed here, a kind of simply drawn capillary bed, okay? And it's got an arterial end, and then the capillary bed's got a venial end. It's the same thing that we have been just looking at here, arterial end, venial end, but I don't see all of the little capillaries just to make things a little bit easier for you to understand. So obviously there's blood in there, and then outside here would be your interstitial fluid. And we kind of talked about a little bit in chapter 19. And if you recall, that's where your cells and tissues would be. We call that interstitial fluid, right? Which is a form of extracellular fluid. And then inside the capillary bed, and obviously we have another form of extracellular fluid, but it's called plasma, right? And again, it's not just in the veinule, but it'd also be in the arterial and also be in the capillary bed. And that's a form of extracellular fluid also. And then the cells themselves that would, be surrounding in this interstitial fluid, inside those cells we'd have, I guess I drew a smiley face. Inside those cells we'd have intracellular fluid, ICF. So just a little bit of reminder what we kind of talked about. So as fluid is moving, from the arterial throughout the capillary bed into the veinal system and then eventually back to the heart, we want an exchange of fluids to happen here. And we're going to look at my diagram when I talk about this, but there's going to be stuff, and when we say stuff, we're going to say bulk flow, fluid that moves out of the blood and fluid that has to come back into the blood. And so typically most fluid moves out of the blood at the arterial end here, which you can see that, it's called filtration. And most fluid moves back into the blood at the veinal end, towards the veinal end, it's called absorption. There's the word net there, and I'll explain why here in a second. This just happens automatically. It's based on a pressure gradient, and the pressure is based on basically blood pressure and osmotic pressure, and that's what we're going to look at next. So a hydrostatic pressure versus chyloid pressure, you're going to see those terms when we look at my diagram. Now, in reality, when you look at this capillary bed, fluid can move out filtration and in absorption throughout the whole capillary bed. But if someone were to say, where do I see the most fluid moving out? Filtration. I see that happening at the arterial end, closer to this end. And I see most fluid coming back in at the veinal end. And we'll call that absorption. There's reasons why, so let's look. By the way, your book goes quite a bit into this concept of called net filtration pressure. You're going to just hear me talk about these two main pressures, hydrostatic pressure and colloid osmotic pressure, which is pressure due to the large plasma proteins. We are not going to then break down these two bigger pressures into their specific names. There's actually two hydrostatic pressures and there's two colloid osmotic pressures. We're not going to I'm going to try to keep things as simple as I can, and we're not going to talk about the numbers or math behind the net filtration pressure, because I think you should be able to understand it without having to do that. So let's go to this figure here. It's one I talked about at the beginning of class that you would need. It's called fluid exchange or bulk flow. That's what this is, across a capillary bed. Let's see if I can make this a little thicker here. It makes it easier to write sometimes. Too thick. All right. And let's use a different color. Let's go blue. All right. So there are two forces that you need to be familiar with. And by the way, down at the bottom, you can see how things are defined, but you'll be able to see me define it here. These are actually called Starling's forces. They're not the same as the Starling, the intrinsic Starling effect in the heart in terms of what comes into the heart has to leave the heart. We talked about that. These are Starling's forces in terms of what's happening at a capillary bed. So this is the arterial end of the capillary bed. This is the veinal end. This would be the simplistic capillary tubes or portion of the bed. And you can see some numbers here. So first thing, you have to understand what hydrostatic pressure is. And hydrostatic pressure is labeled as HP here. Now, if I were to take a little pressure transducer and test the amount of blood pressure at that blue dot right there, I would find out that on average, that blood pressure is about 33 millimeters of mercury. So hydrostatic pressure is a fancy term for blood pressure. That's what it really is, okay? Notice what happens as that blood flow goes towards the venial end. If I took another pressure tube where that blue dot is and I tested the pressure at the venial end, oh, it's less, it's 13 millimeters of mercury. The blood pressure, we call it hydrostatic pressure, but it's basically blood pressure. So in class, I would say, why? Why is that? Why do you think the pressure at the arterial end, blood pressure, hydrostatic pressure, is higher than the pressure at the venial end? Because you can see the numbers, it is. And hopefully one of you should raise your hand. You'd say, well, Professor Finn, the arterial end's close we've just kind of come from the heart. So it's sort of a little bit closer to the pumping mechanism, not right on top of it, but closer. And at the venial end, it's a little further away from the heart. And as blood moves, it moves from a higher blood pressure to a lower blood pressure. So we lose a little pressure as blood is moving along the capillary bed, okay? Now, let's use a different color. Let's use purple. Let's use yellow. Inside the capillary bed, you have larger. Actually, let me do this because it's a little too thick there to do that. I'm going to use a light purple. Let's do that. Let's make this a little thinner. Oops. Give me a second here. All right, there you go. So inside this capillary bed, there are those larger that we learned about, remember, back in chapter 18, the plasma proteins. Now, they're typically too big to leave. Okay, they're typically too big to leave. And they don't, unless there's an issue in your body. So we use the term, these plasma proteins are large. They create a pull of water, kind of a force pressure of water called osmotic pressure. Water follows the solutes. And so notice we can kind of calculate this osmotic pressure, pull of water, and we call it osmotic pressure. But when we calculate it, if I were to use a pressure transducer at any part inside this capillary bed, and I would just measure the osmotic pull of water or fluid, notice the number stays exactly the same because the plasma proteins stay exactly the same. They don't leave the capillary bed. So the pull of water, the pressure of water moving from a high water pressure to a lower water pressure stays exactly the same. Okay, keep that in mind. It's a battle, bulk flow, why I filter at the arterial end, and why I absorb mostly at the venial end is a battle of two pressures, two overall pressures. It's a battle of hydrostatic pressure versus osmotic pressure. So that battle, let's use a different color. You then ask yourself, well, which pressure is higher? Well, in this case, the hydrostatic pressure inside that arterial end is higher than the plasma protein pressure or the osmotic pressure. The pull of those plasma proteins are pulling water. That's why we call it colloid osmotic pressure. Proteins are colloids. Proteins are colloids, same synonymous term. The pull of water back in is about 20 millimeters of mercury, all right? So basically, because 33 blood pressure is higher than 20, we see more fluids actually getting pushed out of the capillary bed because there'd be cells and tissues in here, right? Remember, this is interstitial fluid. We see more substance getting pushed out, filtration, than we do substances pulled in, which would be absorption. So does that mean that there's none of this little none of this little absorption happening? No, it's happening. Let me get rid of it's happening. okay? Just not as much. So I'm gonna actually get rid of that my big plasma protein sign there so that I can, maybe I am. There you go. Notice I'm making my arrows smaller. There's a little bit of absorption going on at the arterial end, just not as much because the plasma protein pressure, the osmotic pressure is only 20. So the pull of water in, where my purple arrows are, you can see the movement of fluid in, is not as much as the 33, the blood pressure push. As blood's coming, through this capillary bed, there is a push of fluid out. And that force, 33, is greater than 20. So that's why you see filtration. That's why you see it at the arterial end. And why do you see, do this again here. Why do you see, I'm going to have to erase my plasma proteins here so that you can see this. Why do you see more absorption back into the capillary bed at the venial end? Well, look at so you see more of this coming in. Why? Look at the plasma protein pull, the osmotic pressure hasn't changed. It's still 20, right? But look, the blood pressure has dropped at the venial end. So now if it's a battle, the winner, oops, just covered my number up so you can't see it. The winner is osmotic pressure now. It's greater than the osmotic pressure, which will help to pull more fluid in, which you can see net absorption occurring, is greater than the push, the 13 millimeters of mercury, the push of fluid out. There's still a little bit of fluid that's going to leave at the venial end, but it's just not as much as coming in. Okay, that's the important part. So that's how you can see how it's a battle of two pressures, because the blood pressure, the hydrostatic pressure, is greater at the arterial end. We see more substances get pushed out, net filtration, than we do net absorption coming in. And because the osmotic pressure now hasn't changed throughout the capillary bed, but it's greater than the 13 millimeters of mercury of blood pressure, and that's why we see the pull, osmosis, osmotic pressure, more fluid is coming in. Water moves from a higher osmotic pressure to a lower osmotic pressure because there's more solids. Water follows the solids, we see more of that. But it doesn't mean that you don't see, filtration and absorption occurring throughout the whole bed. You might look here and go, well, nothing's happening here. Well, yes, it is. But most of the filtration that I would see in a capillary bed happens at the arterial end, and most of the absorption I would see in a capillary bed happens at the venial end. Okay. Let's erase the ink. And let's do this. And then you can see the definitions down here too that I mentioned. So again, if you have questions, please jot them down. All right. And during our Zoom meetings, we will go through them. Now, interestingly enough, you can kind of apply some of this stuff clinically. When you start to think about, here's something that we have to think about, okay? At a capillary bed, interestingly enough, you have probably, and let me show you a picture of this, of the fluid that leaves, okay? So let's say, I'm going to just use a random number. Let's say 100%, let me use of the fluid that leaves mostly at the arterial end, the filtration, of the 100% of that leaves, okay, comes out like this, only about 85% of it goes back in at the venial end, like that absorption. So I've got a little problem, right? 100 minus 85 says that keeps leaving me 15% left over in this interstitial fluid. Now, if that were left there time and time and time and time again, The 15% of this fluid that is left by filtration is that, 100% of the fluid that leaves in filtration, only 85% of it gets back in. That means I'm left over with 15%. It's not reabsorbed or absorbed. Well, I could have a problem. That excess that kept building up could cause tissue fluid buildup, could cause edema. I don't want that to happen. So here's where my lymphatic system comes into play and why there's lymphatic blood vessels. It's one reason why we need the lymphatic system. The other reason is related to the immune system. That excess 15% is picked up, not going back into the vein nodes, it's picked up by the lymphatic system, and then those lymph vessels will actually return it to general circulation. And so we never have edema unless there's issues. And so here's some of the common issues why you would have edema. Normally you don't have to worry about it because the lymphatic system does its job. But if you had a bee sting or something and you had an inflammatory response, then what's leaving a capillary bed is more than what's coming back in. But typically that'll clear up on its own. Somebody gets stung by a bee, they see on their shoulder maybe, and they're not severely allergic, they see shoulder swelling, but eventually it goes away. A little bit more serious, if you had a liver disease or if you were having kidney disease or you were not eating properly, you could decrease the amount of plasma proteins, which then would decrease the amount of pull back in. It would not allow as much absorption to occur and leave excess fluid in your interstitial fluid, okay? And that's not good. These obviously would have to be treated if possible. There's also individuals maybe that have removal of lymphatic vessels like due to cancer, or is what I'm going to show you next, where they're blocked, very rare disorder called elephantiasis, okay? And then because of that, the lymphatic system can't do its job. You might know someone that's had breast cancer, and maybe they had some lymph nodes and lymph vessels removed. Let's say they had breast cancer in their left breast, and they had some lymphatic vessels and lymph nodes removed from their armpit area. And you might notice their arm, their left arm, might seem at times a little bit more, or left armpit area, swollen, edema, kind of edemic, because of, the loss of those lymphatic system vessels. And that can happen. Now this is a very rare disorder called elephantiasis. And if you click, if you're more curious about it, and I'll come back to it, because it is interesting. It doesn't necessarily happen here in the Rochester, Eastern United States area. Yes, I'm not going to be able to get to it. No, that's okay. I don't need to. Oh, boys. No, give me a second here. Oh, shoot. I don't like it when my computer hangs up, and that's not a good thing. That's not a good thing. Let me close this program here. I can reopen it. No big deal. Let me reopen. So elephantiasis is due to basically a parasitic worm that typically make sure I got here, that in tropical areas where, you would be seeing much higher temperatures year round and much more incidences of mosquitoes. So in tropical areas, you could get bit by a mosquito, and that mosquito then could transfer the larvae of this particular parasitic worm into your bloodstream. And eventually that parasitic worm makes its way to, actually I like this better, makes its way into lymphatic vessels and blocks, as it reproduces inside, blocks lymphatic vessels. Now this man has had this disorder for probably a long period of time. You can see the extreme edema in his feet, which you can almost not make out his toes and his legs. Elephantiasis can be treated with an antiparasitic drug. But most likely the individual that you see here probably lives in an area where he does not have access to adequate medical care, or maybe does not have the ability to get adequate medical care in that area, which is so unfortunate. if you research elephantiasis a little bit more, we have and I can't think of the name of it right now. We have a great organization, somewhat affiliated with the United States also, that works on the behalf of these areas in these countries that don't have access to medical care to try to provide as best as possible anti-parasitic drugs when some people don't get them. And some people might not go to a doctor either to know that they've been infected. By the time you see something like this, the anti-parasitic drug can certainly try to attempt to remove any remaining parasitic components out of the person's body. But now the fluid has sat so long, the excess fluid, because the lymphatic system is not able to pick up that excess fluid in these extremities, that most likely this man probably, because these areas, especially that you see around his feet, could become less blood flow, infection, gangrene sets in, and in order so that he doesn't die, would have to amputate. But again, probably lack of healthcare at this point. He just tries to keep these clean as possible. Walking would probably be almost impossible to do here in terms of that. So if somebody did contract this in the early stages, we can take the antiparasitics and never get to a situation like this. But because some of these countries don't have the adequate healthcare, which is so unfortunate, you see things like this based on it. So I just can't remember the name of that organization, but if you Google elephantiasis, you probably will. And hopefully they can keep battling this fight. There's not much you can do to get rid of mosquitoes in some cases. Obviously, there can be environmental control with that. Less mosquitoes means less chance of passing on this parasitic roundworm, but having anti-parasitic drugs, you can help these individuals too. All right, so going to our next kind of like we said, let's see, we'll go back to my whiteboard. If I saved it. Yep. So we have gone through bulk flow and capillary beds. We're now moving into control of blood flow in tissues. Okay. So with most of our control features, control of blood flow in tissues comes down to local control, which is intrinsic control, that the tissues can kind of auto-regulate what they need at their specific time of need based on their metabolic needs. Do we need more oxygen? Because I'm more metabolically active, I need to make more ATP, or I'm not as metabolically active, I don't need as much. And so the tissue itself can kind of control, auto-regulate what it needs. There's also extrinsic factors that control blood flow in tissues, coming from the autonomic nervous system, sympathetic, parasympathetic, and we'll see, and coming from the hormonal portions of the sympathetic nervous system, epinephrine and norepinephrine. Those are outside factors, so we call them extrinsic factors. Now, the local control of blood flow in tissues is kind of complex. We're going to look at just some particular pieces of it, but there are 4 factors that tissues use to decide how much blood flow they need. Degree of vascularization, the myogenic response, local factors released into tissues, and blood flow. Okay. So let's look at these. So these are our 4 local control factors. So here's the first one, degree of vascularization. Now, locally, obviously, more metabolically active tissues like the brain, skeletal muscle, heart, and liver are going to have high vascularity, more blood vessels. Whereas your tendons, your ligaments, your epithelial, like if you have torn your cartilage, you know it takes a little time to repair if you don't have surgical repair because there's not as much vascularization there. Okay, in terms of that. Now, the second way we can auto-regulate or control local blood flow in tissues is kind of related to, we don't really, well, let me say this. The degree of vascularization, this is our first way, besides having, there's certain areas of the body that have already been built to have high vascularity. And it's just going to stay that way for the rest of your life. Otherwise, you die. If your brain doesn't have a high amount of blood vessels at all times, you could die. Your cartilage is never going to suddenly develop more blood vessels. It's built with little vascularity, just is what it is. Now, as part of that degree of vascularization, there is potential to increase the amount of blood vessels in a particular area. So that increases blood flow or perfusion. And we call that angiogenesis. Now in skeletal muscle, that's fine. You can train to build more blood vessels. And if you do that, you will train your body to have your heart beat with less beats per minute. And it can deliver more efficiently oxygen to those tissues. You know, somebody that's really aerobically fit, they have more blood vessels to deliver more oxygen to their skeletal muscle, so their heart doesn't beat as much. You know, somebody at rest that's really physically fit maybe have a resting heart rate around 50. And so as they're exercising, the heart doesn't have to beat as much because angiogenesis. And so if you're working hard aerobically, you would develop more blood vessels, you would deliver more oxygen, you would get the production of more ATP more efficiently. That's a hard word to say. So again, don't ignore my bad English. Angiogenesis, I guess. We want that to happen here in skeletal muscle, okay? We don't want these to happen. So in purple, we're going to gain weight. Some of us do, some of us don't, but inevitably as we age, we tend to put a little more weight on. Adipose tissue, unfortunately, can form new blood vessels. We don't really want that to happen, but it can. And we definitely don't want my coronary blood vessels. It helps a person obviously to not, their heart to continue to function. But when there's more and more blockage in a coronary blood vessel and they start to kind of make new blood vessels to try to bypass that area, it's probably inevitable that that area is going to become completely blocked off. And we really, while it is somewhat good to begin with, it's something that we don't really want to think about happening in our bodies because we don't want the blockages to occur, but it can based on that. All right, so let me see here. So now remember, we're looking at 4. The first one I just covered, degree of vascularization. Now the next local auto control, auto regulation is going to be the myogenic response. Now, a tissue, remember we looked at the capillary beds, and myogenic stands for muscle. The smooth muscle in a blood vessel can keep blood flow relatively constant. So if blood pressure were to rise and more blood enters an arteriole, to keep that blood flow more constant instead of turbulent, the arteriole will stretch somewhat and smooth muscle will contract, and that keeps, even though there's a higher amount of blood pressure coming in, that keeps the blood pressure relatively the same. Same thing if blood pressure decreases, there would be less stretch, you see more of a dilation, and that would keep blood flow relatively constant. So we don't see this kind of high, low, high, low, high, low, high, low, high, low kind of thing. And we do that with those sphincters that we talked about. So you can see this is coming into a capillary bed. There's those precapillary sphincters. They can react to just simple changes in blood flow like oxygen and carbon dioxide. For example, you can see this figure here that vasodilation of precapillary sphincters will occur when they sense. So as the tissue concentrations of nutrients such as oxygen or glucose, amino acids, and fatty acids were to drop. So if you were more metabolically active, you're making more ATP, right, in a very metabolically active tissue. And so if this were the case, you're using your oxygen to make ATP and you're producing more carbon dioxide. Well, that tissue needs more bulk, kind of more blood flow. And so just like if you want to fill up, this is where we're going to head to here shortly. If you wanted to fill up a bucket or water your flowers very thoroughly, you're going to use a vasodilated hose to do that. You don't need a lot of pressure, but you need a lot of volume. And so when this occurs in tissues that are making high amounts of ATP, we want those sphincters to relax and we want dilation to occur to get me more blood flow here because I need it. Now, blood flow is not the same as blood pressure, okay? Think about your vasodilated hose. You'd want to get more water volume-wise into your flowers and more water into your bucket. But there's times where, some tissues have adequate amounts, the oxygen levels are higher or normal, and the carbon dioxide levels have dropped off, which would be normal. There's less ATP production. So you wouldn't want as much blood flow here. You don't want to waste your blood flow, or we bypass it altogether. And so that's when the capillary sphincters will react, they'll contract, and you get a kind of a vasoconstriction. Now there's also, and your book talks a little bit about this, but we're not going to look. Table 20.3 looks at all the local chemical vasodilators and local chemical vasoconstrictors that can cause what you see in this picture to occur also. But we're not going to look at all those. So you can read on your own. You probably talk about them when you move into your future nursing careers in terms of that. Okay. But, I just pointed out some oxygen and carbon dioxide. Oxygen and carbon dioxide can act, obviously, as a local vasodilator here and a local vasoconstrictor here, but there's other local vasodilators and vasoconstrictors that can cause this to happen. We're just not going to look at them. All right. Okay. Finally, so we've gone through degree of vascularization, myogenic response, local factors. Now our last local auto control is total blood flow. The amount of blood transported through vascular per unit of time. This can increase significantly with exercise. There's obviously different regulatory mechanisms that are involved with controlling it. Depends on the heart and the blood vessels, and we're going to be talking about some of these things shortly. Okay. All right. So now we're kind of moving into our last significant objective mean arterial pressure, and that kind of will lead us into the baroreceptor reflex also. So I'd like you to kind of take a minute. You could certainly pause too, just to, if you want to go back, if you have some of your chapter 19 notes, and start to think about what we talked about with mean arterial pressure, because we did. So I'm going to give you kind of a minute to do that. I want to get my things in order here because this is probably our last important piece, and then we'll look at some of the anatomy very briefly of some of the blood vessels, and that will be it. I'll give you about a minute. All right, let's refresh a little bit and review. Remember, we looked at this equation, mean arterial pressure equals, cardiac output times total peripheral resistance. We haven't talked about total peripheral resistance yet. We're going to. But I can rewrite this equation and expand it by saying if I know cardiac output equals stroke volume times heart rate, I can just write this whole equation out like this. Stroke volume times heart rate, and then add the torque per resistance, which we're going to talk about. Okay. How would we define mean arterial pressure? Really, it's the average aortic pressure during the cardiac cycle. Okay. 1 heartbeat. What's the pressure like during 1 heartbeat? Now, if we were really to measure this, which we can, it's a weighted mean, and at rest it averages about 80 to 90 beats per minute. And we say it's a weighted mean because, remember, in 1 heartbeat, we need to know what happens when the ventricles contract. And we need to know what the blood flow is like when the ventricles relax. Okay. Need to know. So, a lot of times you think, well, when the ventricles are relaxing, there's no blood flow of the heart. Well, theoretically, there's no blood flow of the heart, but there's still blood in blood vessels. There still has to be a blood pressure. You wouldn't want, when your ventricles are relaxing, for blood flow to stop everywhere. It would never, your brain needs a constant supply of blood. Obviously, so does your heart, but so do the rest of your body parts. So, that tunica media and some of those larger elastic arteries helps to keep propelling the blood. It can absorb some of the pressure during when the ventricles are contracting and shooting blood out the aorta into the rest of the blood vessels. And then as the ventricles are relaxing, we get this recoil and it keeps that blood flowing. Now remember, 1 cardiac cycle only takes about 0.8 seconds. Okay, so during the 0.8 seconds for 1 heartbeat, okay, about 0.5 of those seconds is when the ventricles are relaxing diastole. And 0.3 of those 8 seconds is when the ventricles are contracting, systole. Okay. So and this would be at rest. In 1 heartbeat, because remember, cardiac cycle is 1 heartbeat, we spend more time relaxing than we do contracting. And so we say mean arterial pressure is a weighted mean because this number at rest is an average of what the blood pressure would be like during these 2 instances, but because we spend more time in diastole, the average tends to be weighted more towards diastole. Think about when you get your blood pressure taken. Systolic over diastolic. And blood pressure using a sphygmometer and a blood pressure cuff, the top number that you see, the 120 number, is the systolic number. What would blood pressure be like when the ventricles are contracting? And the bottom number is the diastolic number. So S means systolic, D means diastolic. That's what blood pressure would be like during diastole. Notice it drops a little bit because the ventricles are contracting. It doesn't completely go to 0. So because we spend more time on diastole, if I added these 2 together, a normal average resting heart rate, that'd be 200. And if I divided 200 to get an average by 2, that'd be 100. So the mean arterial pressure is not 100. Typically at rest for most humans, it's about 80 to 90. And so because we weight there is an equation you could look at, and you could actually measure mean arterial pressure for folks, but it's not going to be the perfect average. It's going to be a weighted mean because we spend more time in diastole. And so erase all that ink. And by the way, if you're curious, we can, not that you have to know this, but this is a textbook definition of how we find a mean arterial pressure. We've got to know the diastolic pressure, diastolic blood pressure, and we need to know what a pulse pressure is, which we're going to probably talk about when we get to our virtual blood pressure lab. Pulse pressure is calculated by knowing what a systolic pressure is minus a diastolic pressure. So we get that number from that. And we took a third of that. So you can see diastolic pressure is heavily weighted in this. You don't have to do this mathematically, but just give you an idea. Now, typically, when you think about blood pressure, the highest blood pressure is going to be in the larger arteries during ventricular systole, and the lowest pressure is going to occur during ventricular diastole. Okay. Are there differences? In mean arterial pressure, can mean arterial pressure change? Yeah, it can change. When we're exercising, certainly can go up. So it's not going to stay at 80 to 90 all the time. Mm-hmm. All right. Let's move on. So here's kind of where we're going to get into looking at a little bit about what kind of factors can affect mean arterial pressure, and then also what total peripheral resistance really means. That's kind of where we're headed to next. Okay. So we're going to expand this. So remember, cardiac output is the amount of blood pumped by the heart per minute, and we can calculate that by knowing the stroke volume, how much gets pumped per heartbeat, times the heart rate. We talked about that in chapter 19. But what is total peripheral resistance? Excuse me, it's the resistance at which blood must be pumped. What kind of resistance will blood get when it's going along through the blood vessels? That's kind of what total peripheral resistance means. So when you start to think about that, let's look at this. We're going to look at this guy first. I'll get my whiteboard. Actually, I want to leave that. So let's use a new slide. That'll work. So if I were to draw a blood vessel that looked like this versus a blood vessel that looked like that. And hopefully you would say, well, that's smaller, Professor Finn. So we're going to say that blood vessel is and it's kind of an artery type of blood vessel. We're going to say it's vasoconstricted. And we're going to say this blood vessel is larger, it's vasodilated. So at the level of blood vessels is where we're thinking about, in regards to total peripheral resistance. So if you imagine blood flowing along this blood vessel, it's going to hit the sides of its container a little bit more. It's going to encounter more resistance as it's moving from point A to point B. Now that doesn't mean it's going to stop, but when we say it encounters more resistance, we say there's more peripheral resistance, but if we look at all of the vasoconstricted arteries that might be happening in our body at one time, we would probably, we'd probably then say, well, there's more than just one blood vessel, so let's call it total peripheral resistance, okay? Here's the thing, because we're going to relate this back to our equation here in a second. If there's more resistance to blood flow in this vasoconstricted artery, well, you can imagine if you have like a vasoconstricted hose, if you're power washing your house, right? You get more pressure coming out of that hose. Now, if I had to water my flowers or fill a bucket up, it'd take me forever to do that. While there's more pressure coming out of that hose, there's less like volume coming out of that hose. That's what you have to look at. So we say if there's more resistance in the circuit, that leads to a higher pressure direct relationship. So we say there's more mean arterial pressure. But in terms of the volume, the flow, not the pressure, in a vasoconstricted artery, there's less flow. If you try to fill a bucket up really quick, you'd have a problem doing that. And also, I use the analogy of if you had your favorite drink and you want to drink it really fast, you wouldn't use a vasoconstricted straw, you'd use a vasodilated straw, right? So with a vasodilated blood vessel, there's less resistance to blood flow. It's not going to hit the sides of its container as much. It's going to kind of flow pretty fluidly right down the blood vessel there. But with a vasodilated hose, and there's less resistance to the blood flow. That would mean there's not as much pressure in that blood vessel or in the blood vessels that are vasodilated. Think about your vasodilated hose. You change the nozzle so that you get more flow coming out, less pressure, so you don't blow your flowers right out of the bed that you just planted nicely, right? And and we'll talk about now why we'd want differences here next with vasoconstriction and vasodilated arteries. But now you have to think about that the vasodilated hose is not like a pressure washer. There's not a lot of pressure, but it can give us some more flow, more blood flow, more blood volume. So again, if you wanted to drink and get a lot of fluid volume in your mouth of your favorite drink, you're going to use your vasodilated straw. If you wanted to here's another, if you have, one of those water picks, if your dentist said we want you to use a water pick in addition to tooth floss, if you look at the tip of your water pick, the little water flosser, it's very constricted. What does it do? It sends a high stream of pressure out to clean in between your teeth. If you had a vasodilated, water pick, you'd never get in between your teeth to blow out that stuff that your dentist is wanting you to. You get a mouthful of water instead very quickly, okay? Now, in our bodies, this is important. So here's something to think about. There's times, obviously, when you're exercising and those skeletal muscles need more blood flow to pick up more oxygen to make more ATP. So if I look deep inside my skeletal muscles at the time of exercise, I'd see that some of the blood vessels are very vasodilated because I don't need pressure at that time. I need more bulk. I need more blood flow going in, okay, to provide more oxygen and nutrients. But that blood has to get to the skeletal muscles rather quickly. When you start exercising, you can't wait, for your blood pressure to, it starts to increase, but you need blood vessels to route that blood very quickly to the areas that need the most flow. So here's an example. When you're exercising, your digestive tract doesn't need much blood flow. It's not as important. We're not going to completely shut the blood flow off in it, but many of the blood vessels in your digestive tract would be vasoconstricted. Why? We don't need as much flow there. We're bypassing some of those tissue beds of the digestive system. We're constricting them, remember, those capillary sphincters, so we can speed up the speed of the blood flow or the pressure. And then those vasoconstricted arteries can reroute the blood into the places like skeletal muscle that need the blood flow the most. And then by the time I get to the skeletal muscle, local blood flow will be happening and we'll see more vasodilated blood vessels. Now, there are times in your body where you do have vasoconstricted areas and vasodilated areas. That's fine. There should never be a time in your body where you have all vasodilated areas. That's cardiovascular shock. That can be very detrimental, obviously, to a person because you can't generate enough blood pressure to do that. And I think I do have a video that I'll alert you to that you can watch on your own in terms of that. This was also another analogy of my house, my 3-story house. When I'm taking a shower on the 3rd floor, and I got that nice flow of water coming in, The body is the same way. When areas are vasoconstricted, they're allowing, more pressure to be generated, but there's less flow going to the tissues so that we save it for the tissues that need it the most. Well, in a house, sometimes that doesn't work with old pipes and such. You can't adjust the size of the pipes. So as I'm taking my shower, I'm getting that nice flow. When I'm turning faucets on and off and flushing toilets, that can, because I can't vasoconstrict, your toilet or your faucet to give me more flow. That's when you stand in the shower, you don't get any water on you, and you go, darn it, they flushed the toilet. Now our body doesn't work like that, but it's kind of interesting in terms of seeing that. So now we're going to see that blood vessel size does have an effect on blood pressure as well as stroke volume and heart rate. And that's where we're headed to next. So read this slide. There's the expanded version. It still means the same thing. We've just expanded cardiac output. Okay? Now, anything that affects either of these three can affect mean arterial pressure. So, here's the thing. If any of these three increase, if stroke volume increases, if heart rate increases, and if total peripheral resistance increases, and when we say total peripheral resistance increases, we're saying there's more vasoconstriction in the network, what I just showed you. It's a direct relationship. It causes mean arterial pressure to increase. Okay? Versus, which is a different color here, if any of these three decreases, stroke volume decreases, if heart rate decreases, if total peripheral resistance decreases, which means there's more vasodilation in the network, then mean arterial pressure decreases. Direct relationship. Now that's the easy part to understand. And that's one thing when you're learning about this, you've got to know in your mind if there's, for example, if there's less blood leaving per beat, stroke volume, if there's less heartbeats, if there's more vasodilation, which leads to less total peripheral resistance, that will decrease your blood pressure. Or vice versa, if any of these three increase, that will increase your blood pressure. You've got to know that. But then that's going to get tied into the baroreceptor reflex. There's times where our blood pressure increases, if we're exercising or anxious, etc. We don't want it to stay elevated all the time. No, again, at rest, we want our blood pressure, and again, this is how we would manually measure it. We want it to be below 120 and below 80. 120 systolically, below 80 diastolically at rest. And so if you're exercising, obviously the systolic number is definitely going to go up. Diastolic changes very little, typically if you're in decent shape. And so we've got to have a negative feedback mechanism that when we decide to stop exercising or if our anxiety ends, etc., we want our blood pressure to go back to normal. And we call that the baroreceptor reflex. And it works vice versa too. What if your blood pressure is lower than normal? And I'll give you some instances of how that can happen. Well, we want your baroreceptor reflex, which we're going to see as part of the extrinsic part of control, to go back up to normal. This is very important. And within seconds to minutes, you don't even notice this happening, but your body is using a negative feedback mechanism with the help of your extrinsic factors to bring you back to normal. So not only do you have to understand the direct relationships, but then you're going to have to think about what normally would happen if these three decreased and what normally would happen if these three increased. That's the first part of the idea. The second part is now thinking about this is occurring, but we want this to occur. Okay, negative feedback. Or this is occurring, but we want to get back to normal. What would happen? And that's where it can you really have to understand this. We'll see next. All right. So I think we saw a similar picture like this in chapter 19. The green that you see down here is sympathetic nervous system, pre and postganglionic fibers, innervating the SA node, the ventricular myocardial tissue, and the smooth muscle of arteries and veins. Notice it can control heart rate, it can control stroke volume, and now the sympathetic nervous system has control total resistance. We talked a little bit about this in our chapter 19, but let's look at it very briefly again. When the sympathetic nervous system predominates, it can control all three pieces of this mean arterial pressure or blood pressure equation. It can tell the heart rate. If you're in a fight or flight situation or exercise, it can tell the heart rate to speed up. Let me use a different color here so you can see it. Because it controls the SA node. When the sympathetic nervous system predominates, it can tell your ventricular myocardial tissue, hey, contract with more force. So you force more blood out of the heart. Yep. Increase stroke volume. And then it can tell your arteries and veins, for the most part, not every single one of them, but for the most part, some of them, depending on the areas of the body that we would need this to occur, to increase your level of vasoconstriction, and that would increase total peripheral resistance. And guess what? When the sympathetic system predominates, so the sympathetic system can control stroke volume, it can control heart rate, it can control total peripheral resistance because it can control the smooth muscle on arteries and veins. All of those features will increase mean arterial pressure. Sympathetic system is very important to do that. It's very widespread effects. And the sympathetic system, obviously, in addition to having these wirings to these areas, epinephrine and norepinephrine coming out of the medulla can also bind all these areas, obviously the SA node, the ventricular myocardial tissue, the artery smooth muscle, the venous smooth muscle that's controlled by the sympathetic nervous system is going to have receptors there that the sympathetic neurotransmitters can bind to, and the hormonal part, epinephrine and norepinephrine, can bind to. By the way, I'm assuming that you already know this. So let me get rid of this. There is, in test tube materials, there's a PowerPoint called Brief Review of the Autonomic Nervous System. And when you open that PowerPoint up, I also did a recorded lecture. Let me go to my Connect section here. And this is similar how you guys would, oops, I don't want to do that. This is what you would get to your student homepage here. And when you go down to the recorded lecture feature, this is the easiest way to get to it, maybe. I done did it, didn't I? Let me see if it's going to load for me. Nope. Double nope. All right, so let me see. Hopefully my internet didn't die here. All right. Now you're not going to have to go to the recorded lectures like this. Remember, you can go to McGraw-Hill Resources and see the same thing that you'll see here. And then your view will look like this. Yay, there it is. And you go down to the bottom here and click recorded lecture. And right there, brief review of the sympathetic and parasympathetic autonomic system. Now, you should have learned this in Bio 144 right at the end, but I know from the spring of 2020, This has been kind of a difficult transition with the COVID virus. And you kind of went in the middle of being face-to-face and then had to learn online when you weren't really knowing that was going to happen. So I've previously recorded this video, and it's probably more important than ever. If you didn't get a good idea of the sympathetic and parasympathetic system and how it works anatomically and physiologically, you might have to watch this. Now, I'm not going to ask you questions from this, but in order to understand what's happening here with the receptors and a sympathetic involvement, you might need to, okay? So just an idea for you. Now, what about the parasympathetic system? I can see it, let me do it this way. I can see it in blue here. I notice it doesn't have as much connections, right? And as we talked about very briefly in chapter 19, stroke volume times heart rate. Times TPR. Now, the parasympathetic system is not as involved in controlling blood pressure in terms of these three components. It can control heart rate only, because you see there is a pre and postganglionic neuron kind of innervating the SA node that can control heart rate. But I don't see any blue fibers controlling the ventricular myocardial tissue or the smooth muscle of arteries and veins. So the parasympathetic system can only control heart rate. That's it. And when it does control heart rate, it tells the heart rate to slow down back to its normal speed. So maybe after you're exercising, the sympathetic system is predominating. The parasympathetic system will tell the heart rate now it predominates, and you get more, obviously, neuronal activity. Instead of the green fibers, you get more neuronal activity on the blue fibers. And the parasympathetic system has receptors on the SA node that will then tell the SA node, don't speed up, slow down, and that will tell the blood pressure to slow down. Now, stroke volume and total peripheral resistance will go back to normal, not because the parasympathetic nervous system are telling them directly to, because indirectly they don't have that sympathetic control that was predominating. So, remember, when the sympathetic system predominates, stroke volume increases, total peripheral resistance increases, but now it's going to kind of go back to normal. We'll see that with the baroreceptor reflex. Pretty important concept there. Now, before we finish up with this baroreceptor reflex, we have to also really understand that the amount of blood returning to the heart, venous blood pressure from the veins, it can affect blood pressure. So if you just use this kind of idea, if you're exercising and more, I'm going to call venous return VR, more VR, venous return, we're coming back to the heart. Well, think about from chapter 19, okay? That's going to fill the heart, the ventricles, with more blood. Remember, that's called end-diastolic volume. More venous return, more blood filling the ventricles during diastole before we're going to eject. So if we fill more, we talked about Frank Long's law of the heart. It's Frank Strong's law of the heart. More sarcomere stretch, more cross bridges are forming. More contractile force, so that increases stroke volume. More blood per beat can lead to more blood per unit of time. And guess what? More blood per unit of time can lead to more mean arterial pressure. And by the way, if we don't expand the equation, the same principles would result if cardiac output increases, mean arterial pressure increases, right? If cardiac output decreases, mean arterial pressure decreases. So if there's less blood flow coming into the heart, then each of these would be less. The problem with venous return is there's hardly any pressure in the veins. And in the big veins, the vena cava, the superior vena cava, the blood pressure is almost zero. And that's weird. So you're like, well, blood's supposed to flow from a higher blood pressure to a lower blood pressure. How's blood get back to the heart then, especially from your lower extremities? Venous pressure doesn't have, it's low, it's not pulsatile like arterial pressure. It doesn't have this pumping mechanism like the heart. Well, how do I get it back? Well, I depend on three things. The pressure gradient is not significant, but the skeletal muscle pump, the respiratory pump, and something along the autonomic nervous system called venomotor tone. Let's look at those. So just by simply moving, when you walk, these are your calf muscles, by the way, it's helping to push blood up towards the heart. And then when it's kind of pushing here, and when it stops and maybe the muscles relax, blood doesn't flow all the way back down because you have the valves. Now, when blood gets into your abdominal cavity, how does it make that last little trek up into your thoracic cavity? And again, in this, obviously you're thinking about blood coming from the superior vena cava, it's moving down, so I don't have to worry about it too much. But blood moving up, the inferior vena cava, I do. And so with the help of breathing, the respiratory pump, when you are inhaling, and we're eventually going to get to this, it drops the pressure in your thoracic cavity. And there's more blood pressure in your abdominal cavity, so that helps the blood to go against gravity. Okay, it's kind of interesting. So we have these two pumps that are working to help bring blood back towards our heart. But we also need a little help from our autonomic nervous system, in particular, the sympathetic nervous system. So the sympathetic nervous system can cause venous veins, smooth muscle, to constrict, and that can help increase pressure in the veins, and that forces more blood to the heart. So we had been talking about arteries, so now you're seeing this with veins. You're seeing a little bit more of vasoconstriction, which would increase the resistance of blood flow into the veins and increase the pressure to push it back towards the abdominal cavity and the heart. Now, we've already talked about resistance with blood vessel size, vasoconstriction versus vasodilation. Total resistance can also be affected by the thickness of blood. We don't want blood thickness to change. If you were to get severely dehydrated, you wouldn't want that to happen, but it can, and that could create issues in our body. We wouldn't want that to happen, and we can't really change blood vessel length. It's not going to suddenly go from a small blood vessel to a longer blood vessel, but we do have smaller blood vessels and longer blood vessels already built in our body, and that can affect resistance. We mostly are going to focus on vasoconstriction and vasodilation, which we just talked about. So you can kind of review that slide. Now, as we get into some of our hormones as part of the urinary system, like there's ADH, which is also called vasopressin, and angiotensin II. Briefly mentioned in this chapter, but we're going to talk more about it in our urinary system. That can affect blood vessel size and blood pressure also. So, our kind of last little bit. We've kind of have led ourselves now into the baroreceptor reflex. And before I go into that, this is part of regulation of blood pressure and blood flow. You've got to have your blood pressure be kept in a proper range, high enough to maintain tissue perfusion, not so high to damage blood vessels, and not so low to not get blood flow where you need to go. It depends on cardiac output, that depends on total blood resistance, and that depends on your blood volume too. Your nervous system, your endocrine systems can regulate that. Now we're going to look at what we call short-term regulation, which is the baroreceptor effect. This is quick changes in blood pressure, meaning minutes to seconds. If you're, or in a recovery situation after exercise, your blood pressure changes rather quickly. If you exercise and your blood pressure and heart rate elevate, right, it doesn't take 3 hours or 3 days for your blood pressure to get back to normal. Now, we do need longer term regulation of blood pressure. This would be hours to days. Your book finishes, chapter 20 kind of finishes with this. Several of these hormonal regulatory systems, the RAS system, the ADH vasopressin system, and the ANP or ANH atrial natriuretic system. They're presented, so please pay attention very closely. They are presented in chapter 20. We are not going to cover them in detail and for testing assessment until the urinary system, okay? We are not going to cover these in detail and for assessment until the urinary system. So you're going to see these again, okay? But this will not be on test 2. So I'm just going to put no test 2. I don't want to cross them out because we're still going to learn them, but I won't cover this on test 2. Okay. No test 2. We're going to, but not yet. No test 2. Why are they presented here? Well, to give you a little bit of a taste, your book goes a little bit more involved in this chapter. I'm going to cover them more in detail when we get into the urinary system, because you're going to see that the urinary system not only controls fluid and electrolyte balance, but also can be involved with blood pressure regulation because of blood volume regulation. So no test 2. I think that's it. Yes. So those slides that I just did, which are basically slide 44 through 48, not on test 2. Okay. Let's see. Back to here. Let's talk about the baroreceptor flex. To do that, we need to talk about, well, how does my body know when to tell the sympathetic nervous system to predominate in this regulation of blood pressure, and when the parasympathetic system should predominate during this regulation of blood pressure? And that, you've got it. With any negative feedback mechanism, if you're going to return something back to normal, opposite change, you've got to have a control center. And our control center in this case is the cardiovascular center. There it is. So that area within the medulla oblongata is called the cardiovascular control center, cardiovascular center, but it contains 3 parts. 1, 2, 3. Well, actually, make this a little smaller. One, 2, 3. It contains a vasomotor center. It contains a cardioacceleratory center. It contains a cardioinhibitory center. Right? Now, let's start with the cardioacceleratory center. Well, first of all, let me say this. The vasomotor center is what controls total peripheral resistance in terms of the size of your blood vessels. And that is, in terms of the extrinsic control, is dedicated to let the sympathetic nervous system carry that out. But that vasomotor center has to kind of communicate with the sympathetic system. And so the parasympathetic system is not involved with total peripheral resistance. There is no, as we talked about before, neuronal connections or neuronal ganglionic fibers that are innervating those areas. Now you can see the vasomotor center right here controlling the size of blood vessels. That comes from the vasomotor center. Now the interesting part about that is, I did mention this a little bit. I don't want to get too far involved. I'm going to mention a little bit. Not all blood vessels are going to vasoconstrict at the same time, and we don't want all blood vessels to vasodilate at the same time. We want kind of a mixture. Those areas that need more blood flow will have, especially on a consistent basis, like blood vessels to your skeletal muscles or coronary vessels, they're going to have special adrenergic receptors, and again, watch that video if you can't remember what that meant, beta 2 adrenergic receptors, that sympathetic neurotransmitters like norepinephrine or hormone from the sympathetic nervous system like epinephrine and norepinephrine can bind to, and they would cause a smooth muscle to relax and vasodilate. But then there's areas where we need vasoconstriction to occur, and blood vessels, immediately, like my brain has to say, time to vasoconstrict, time to vasodilate. And that's coming from the cardiovascular control center, particularly the vasomotor center. So the sympathetic nervous system can also cause some blood vessels to constrict at the same time. And because those blood vessels have alpha 1 adrenergic receptors. And so you kind of talked about this in your Bio 144 class. So that's kind of how we can see different areas of the body having more speeding up blood flow, and then different areas of the body having more bulk flow coming out because of the different adrenergic receptors. There are different reasons for it. Okay, so that's TPR. The cardiac-oscillatory center is part of our cardiac center. Now, the cardiovascular control center has three things that are involved with it: a vasomotor center and then a cardiac center. But the cardiac center is broken down into a speed-up center, cardiac-oscillatory center, and a slow-down center. The speed-up center, guess what, is controlled by the sympathetic nervous system. Well, I should say, it's not controlled by the sympathetic nervous system. The sympathetic nervous system does its job. So when the cardiac-oscillatory center is turned on, the sympathetic nervous system will then, the wiring, the pre and postganglionic neurons will then control, we just talked about this, the SA node. You can see the green wires going to the SA node here, right there. And the green wires can also go to the myocardial tissues here. So remember, the sympathetic system can control stroke volume and heart rate. How does it do that? It gets a message from the cardiac-oscillatory center. It says, okay, sympathetic system, you predominate. And when you do, we can speed up heart rate. We can speed up stroke volume. If the vasomotor center is more active, we can increase total peripheral resistance in some places. Okay. There, that all can increase mean arterial pressure overall. There are some areas where we need a constant supply of blood flow, especially the coronary vessels. So when the sympathetic system predominates, we don't want the coronary blood vessels to constrict. Our heart would not get enough blood flow. So that's why they have different receptors and why they would vasodilate. But when the sympathetic system predominates, most blood vessels with alpha-1 receptors are going to have a vasoconstricted effect. It will increase total peripheral resistance to get blood flow to the areas that need it, skeletal muscles and the heart and brain, and you would see different receptors there that when the sympathetic system is predominating, it tells those blood vessels to vasodilate. Now notice I haven't said anything about the parasympathetic system yet, because remember, it can only control heart rate. So there's a yellow kind of parasympathetic system, pre and postganglionic neuron. You can see it's only kind of controlling the, not only SA node, both systems control SA node and AV node, but the SA node is your master pacemaker. So let's just use that. Well, what if we want to again, the sympathetic nervous system, all these things are increasing blood pressure, mean arterial pressure. Well, what if we want blood pressure to go back down to normal? Well, then the cardioinhibitory center will be active. It'll tell the parasympathetic nervous system, hey, you need to slow the heart rate down, and guess what that does to mean arterial pressure? We talked about that, right? Just as those three things can increase mean arterial pressure in blue, the parasympathetic system can only control heart rate. So that when the cardioinhibitory center is activated, the only thing that's going to happen is from the parasympathetic side of things is heart rate is going to drop and mean arterial pressure will drop back to normal. We're going to talk about stroke volume and total peripheral resistance indirectly here in a second. Now, with any negative feedback mechanism, how does the cardiovascular center know when to activate its cardiac-oscillatory center, when to activate its cardioinhibitory center, when to really activate its vasomotor center? How does it know? With any negative feedback mechanism, you've got to have sensors. So this is your control center. But your control center needs to know what's happening. So that's why you have these receptors or sensors around your body that can sense blood pressure, or that can sense the level of oxygen or carbon dioxide. Baroreceptors in your carotid sinus, as well as your aortic arch, can sense blood pressure. We're going to talk about those next. The baroreceptor reflex. Your chemoreceptors in your carotid bodies and your aortic bodies can sense oxygen and carbon dioxide. Right? You also have some in the baroreceptors right here. So that sensor information gets sent up here. You know, if the blood pressure is too high to begin with, this is where we're headed to next, and we want to lower it, then you want to lower it back to normal. This is where I would ask you in class, what would be active in that cardiovascular center? Well, the cardioinhibitory center would be active. These two wouldn't, because these two typically are going to cause the sympathetic nervous system. Remember, I also have to have effectors. My effectors are the motor output, the sympathetic nervous system and the parasympathetic nervous system, to make that opposite change. So to make the opposite change, I'd want the parasympathetic system to predominate here. I'm going to abbreviate. So that this is what we're going to look at next, so that my blood pressure drops back to normal. And the only thing is to tell the cardioinhibitory center be active, which will tell the parasympathetic nervous system to be active, heart rate will drop, blood pressure will drop. What vice versa, if I want, for example, the opposite thing to happen. What if my blood pressure is lower and I want it to increase back to normal? The sympathetic nervous system is in charge of increasing blood pressure. So my vasomotor center and my cardioacceleratory center are going to be active. And remember that with the cardioacceleratory center, my heart rate can increase, my stroke volume can increase. My TPR can increase for the vasomotor center. All of these things are increasing blood pressure. That's the second way you're going to have to start to think about this. And we're putting it all together so you can see it next in this, this figure. So, moving on to our last thing. The baroreceptor reflex. Now to look at this, I want you to have these out in front of you. And let's see if I can make mine a little bigger here. Yes. And we're going to look at the side that says page 524 first, with the mean arterial pressure at the top saying decrease. So go ahead and find that. I'll give you a couple seconds to do that. All right, so let's look. This is short-term regulation of blood pressure. This can get you back to normal within seconds to minutes. All right. So this is set up as a negative feedback mechanism, as we talked about before. You look at the figure's name. It says the baroreceptor reflex, the response to orthostatic hypotension. What does that mean? Well, it means orthostatic means upon standing. So when you go from maybe a reclining, you're going to see my great artistic work here, from a reclining position to a standing position. Many of you don't even notice this, but your blood pressure will actually drop. The orthostatic part is going from reclining to standing. That's the orthostatic part. Hypotension means your blood pressure drops when you do that. Now, for some of us, our blood pressure drops enough that we feel a little dizzy and we can pass out. And that means your blood pressure is already low to begin with. Maybe instead of 120 over 80 at rest, you're like 80 over 60 at rest. And so when I make you stand up quickly like this, you get a little lightheaded because all the blood is gravity-wise is moving down very quickly. And for some reason, your baroreceptor reflex doesn't respond as quickly, and your body will end up, the brain not getting enough blood flow. And guess what happens? It's an automatic mechanism that makes you pass out. You go back like this, the blood flow will get back to your brain, but no one wants to pass out because you could hurt yourself on the way down. So some of us do have that issue, but let's not talk about that right now. Let's talk about what normally should happen, right? There, I just took his body away from him. Let's get him back. Give him a neck. There we go. All right. So this is the trigger, a drop in blood pressure upon standing. And again, in a negative feedback mechanism, that's what this sheet is showing from a different book. You have sensors, receptors. So notice, look at this very carefully. That's the cardiovascular control center in the medulla oblongata that we just talked about. How does that center know that the blood pressure has dropped? These baroreceptors, we're not going to talk about the chemoreceptors, but these baroreceptors in your carotid arteries and your aortic arch detect the lower blood pressure. How do they tell the cardiovascular control center what's happening? They have to use action potential language. So you see decreased firing of carotid and aortic baroreceptors. What does that mean? That means decreased electrical activity, decreased action potentials. That's what that means. That the brain, the cardiovascular control center, I'm just going to draw a fictitious action potential here, just the depolarization and repolarization. It's going to see very infrequent action potentials, saying there's less blood pressure. That the frequency is decreased action potential frequency gets to the cardiovascular control center, and the cardiovascular control center goes, hey, the blood pressure is lower than normal. Now, what is lower than normal? Well, not way lower than normal, but let me give you this idea. I'm going to get rid of this so I can use a different color here. And normally, when your mean arterial pressure is in this range, your baroreceptor reflex can work efficiently. So let's say I stood up suddenly and maybe my mean arterial pressure was 90 and it went down to 80, okay? Or in some cases, for some of us, it could go a little lower, but not much, maybe 70. Well, I can't keep it there all the time. I'm not getting enough blood flow in my brain. So those carotid and aortic baroreceptors would fire less frequent action potentials. And then the cardiovascular center would say to itself, okay, I need to get blood pressure back up to normal. How might I do that? And so now you're thinking about this equation. How do you get blood pressure back up to normal? We've learned so far that if I'm going to I've got to increase my mean arterial pressure. If I'm going to increase my mean arterial pressure, I need my stroke volume to increase, my heart rate to increase, and some of my blood vessels to get increased total peripheral resistance. All three of these things will increase mean arterial pressure, blood pressure. Guess what system can do this? We've already learned about it. The sympathetic system can do this. So these are your effector responses. You've got your trigger, you've got your sensors. This is your control center. in the negative feedback world. Not control, control. See here. I have to probably get rid of some of this thing. And then these are your effector responses. Okay. And what the effectors do is they will actually do the job to bring it back to normal. I'm going to have to erase some of this because it's getting kind of messy. So let me do that. Get rid of this and get rid of my... So here we can see the sympathetic nervous system in action. I think I'm going to use darker color here. Now the arrow says the sympathetic output is increasing. That means it's, sorry, I'm going to sneeze. Excuse me. That means the sympathetic system is being told you need to be more active. You're going to predominate. We're going to go back to how the, sorry, sneezes. Excuse me, allergy season right now. How the cardiovascular control center controls that, because we just looked at it. But before we do that, I want you to see how the sympathetic system causes stroke volume, heart rate, and total resistance to increase. You can actually see it here. It tells most, not all, most arterial arteries and veins to contract their smooth muscle and vasoconstrict. When you vasoconstrict, guess what you do? For most of the blood vessels, not all, you increase your total peripheral resistance. And if you do, guess what? You increase your blood pressure. That's what we wanted to happen. The sympathetic nervous system also has predominating fibers that are leading to the ventricles. And when they release their neurotransmitters or hormone binds to those receptors on those ventricles, you're going to increase your stroke volume or your force of contraction. When you contract with more force, you get more blood pressure. And finally, the sympathetic nervous system has controlling fibers to the SA node that will increase heart rate, which will increase mean arterial pressure. Now, the combined facts of stroke volume and heart rate increasing increase cardiac output. But look at, there's your equation. Total peripheral resistance, stroke volume, heart rate. Heart rate, stroke volume, total peripheral resistance are all increasing. That's how my blood pressure increases, and then I don't pass out. Happens within seconds to minutes, snap of the finger. Now, let's go back to my question. How does the sympathetic system know to tell the heart rate and the stroke volume to increase and to tell some of the blood vessels to constrict? Let's look. We just looked at it. So in the case that you just saw, we were just looking at it, the cardiovascular center would activate the vasomotor center, which would cause most blood vessels to do this, vasoconstrict, increase the total peripheral resistance, and it would activate the cardioacceleratory center, which would increase the heart rate, increase the stroke volume, all sympathetically controlled. Okay, so if I were to, I'm going to erase all this so you can see a little better here. I might erase all this. I might not. Stuck. I see we're stuck. Give me a second here. So how does the sympathetic nervous system know how to do this? Well, the baroreceptor that are the sensors that are telling the cardiovascular control center that blood pressure has dropped, that will activate the vasomotor center and the cardioacceleratory center. And that's what causes the vasomotor center will cause the total peripheral resistance to increase. And the cardioacceleratory center will cause the stroke volume and the heart rate to increase. So that's how the total peripheral resistance increases, because the vasomotor center, and the cardiovascular center tells the stroke volume and the heart rate to increase. But this is where, and when I'm saying that, the cardiovascular acceleratory center has sympathetic fibers that are going to be communicated to. And those sympathetic fibers will then tell stroke volume and heart rate to increase. And the vasomotor center has sympathetic fibers that are associated with it that will then tell the blood vessels, most blood vessels, smooth muscle, constrict, increase total peripheral resistance. Now, I'm going to erase everything because there is a kind of a little caveat to this. If your blood pressure were to go dangerously low, if you had some kind of issue where there was hypovolemia, which would mean you either lost a bunch of blood from your blood vessels, and that made your hypovolemia means your blood volume decreases, or you lost a bunch of water from your blood vessels, from your plasma, and it went below the normal 80 millimeters of mercury, then we've got issues then. And your short-term baroreceptor reflex can't work. This can't save me. I may need, if it's more moderate or severe loss of blood or dehydration, I might need to call and to get myself into a hospital setting to replace the blood or replace the water. This cannot make blood up, and this cannot make water up. You might have to be given that. So that probably makes sense to you in terms of that. Let's look at the flip side of this. Now, whoopsie. And that would be what happens to the opposite effect. What if your blood pressure were to increase above normal, like maybe I'm exercising? Okay, you can see the response to increased blood pressure. Well, maybe I'm exercising here. There's my trigger. Okay. There's my receptors or sensors again right there. Now, those baroreceptors are going to detect higher blood pressure, maybe because I'm exercising, and I want the blood pressure to return back to normal, negative feedback. So now here's the difference. How do they tell that cardiovascular control center what's happening? They will increase their electrical activity or increase the action potential frequency. That's how they say blood pressure is too high, cardiovascular control center, so you'd see more frequent action potentials. Okay. Now your cardiovascular control center is your control center in the negative feedback mechanism in the medulla. And guess who gets told to predominate here? The parasympathetic nervous system. This would be the cardioinhibitory center that's now activated. Not the acceleratory center, not the vasomotor center. The cardioinhibitory center would be activated. Okay. When the cardioinhibitory center is activated, that means it's going to talk and tell the parasympathetic system it's going to predominate. And as we talked about before, the only thing the parasympathetic nervous system can control is heart rate as part of that cardioinhibitory center. I'm going to find a color that contrasts here. There we go. So the only thing the inhibitory center can tell the parasympathetic system to do, because the parasympathetic system is going to increase its predomination, you're going to see more electrical activity, is it can only tell heart rate to drop. It can't do anything directly to stroke volume and TPR, but we're going to see what happens with them. So that helps to drop blood pressure. But when there's less, so remember, with the autonomic nervous systems, one system will predominate over the other. If the parasympathetic system is more electrically active and being told to predominate, it's like a volume control. Imagine you're turning the volume up on your parasympathetic system radio, and you hear it louder. But your sympathetic system radio right next to it, when the parasympathetic system predominates, you have to turn your sympathetic system radio down. Now, before we looked at the sympathetic side of things, if the sympathetic system radio is predominating, you turn that up, but the parasympathetic system radio gets turned down. Both autonomic nervous system arms cannot be active at the same time. one will predominate and cause more electricity on those neuronal ganglionic fibers than the other. So in this case, your parasympathetic system is more predominating. It's going to tell the fibers of the SA node, hey, slow it down. Now, what about, you can see, and let's use, you can see green here. Why is this happening? Vasodilation, decreased stroke volume, decreased, well, vasodilation was causing decreased total peripheral resistance. Can't really see that. Let me. So decreased TPR. Why are these two things happening? Is the parasympathetic nervous system telling the stroke volume directly, the ventricular myocardial tissue, hey, contract with less force, less stroke volume? Is the parasympathetic nervous system telling the arterial smooth muscle of most blood vessels, not all, but most, not many, not the majority of our blood vessels, but enough to vasodilate? Relax your smooth muscle, vasodilate. No, it can't. It cannot. The parasympathetic system cannot directly control stroke volume and cannot directly control total peripheral resistance. It doesn't have fibers to the smooth muscle of blood vessels or to the ventricular myocardial tissue. It doesn't. The only thing that the parasympathetic system can control is heart rate. That is it. That's the only thing it can control. But then why is this happening right here with my black arrows? Because when the sympathetic radio or the sympathetic fibers are told to have less electricity on them, things go back to normal. Remember, when the sympathetic nervous system predominated, let me pull up my sympathetic side right here. When the sympathetic nervous system predominated, stroke volume increased, peripheral resistance increased. Well, now we're going back to the normal state. When the sympathetic nervous system is not predominating, the normal state will happen. They'll get the ventricular myocardial fibers will get less electrical activity from the sympathetic nervous system. They'll go back to their normal lower amount of contraction, less stroke volume. Most of the blood vessels that were constricted will go back to their normal, because they're not getting a sympathetic signal to constrict. They'll go back to their normal vasodilated position. Okay, so those black arrows that you see there, the parasympathetic system that I've highlighted in green is not doing that. It's simply because you turned down the sympathetic radio. The bottom line here is that all of these things, decreased TPR, decreased stroke volume, because we've turned the sympathetic radio down, and the direct result of the parasympathetic nervous system predominating, decreased heart rate, will decrease mean arterial pressure. Once again, this short-term mechanism, this is short-term regulation, once again, of blood pressure when your blood pressure increases above normal, but your blood pressure is within this range. So maybe it goes up to 120, 130. This is mean arterial pressure. To get it back down to its normal mean arterial pressure, this happens. These are the effector responses that occur because the sensors told the cardio, control center, hey, tell the cardioinhibitory center to get predominating, and that will tell the parasympathetic system to predominate. That will create changes directly with heart rate, to drop mean arterial pressure, and then the sympathetic normal will occur because the sympathetic nervous system has been turned down. So those areas, ventricular myocardial tissue and some of the blood vessel, smooth muscle, kind of goes back to normal. And that's why we see those things right there. What if your blood pressure goes above the normal range? This would be hypervolemia. This short-term regulatory mechanism, the baroreceptor reflex, can't return you back to normal in most cases if it's moderate to severe. If your kidneys fail, or if you were to drink too much water too quickly, there's other reasons why you can get into hypervolemia too, but you would need, most likely if it's moderate to severe situation, 911 help to get we got to remove fluid from you. If your kidneys are failed, to get you back to normal, maybe dialysis, that type of thing. Okay, so there's your baroreceptor reflex. Get rid of that. And remember, I had talked about the long-term regulation of blood pressure. We're not going to go into that for this test. I already mentioned that. So these slides, you will not see any information on them for test 2. So a little bit of a kind of a summary with 2016A, 2016B, 2016C. I'm pretty sure, because we're going to do a video on blood pressure, but let me just make sure here. I put a video. Yeah, this will talk about hypertension for you. Hypotension, which is below normal blood pressure, we talked a little bit about. This will discuss hypertension, and so you can watch that for the clinical kind of feature of that. Yeah. So the rest of the slides pretty much are anatomical definitions of blood vessels. I will be making a separate lab video for that. Again, you're going to see some of these anatomical features on a lab on a lecture exam. I will set up I have set up a PowerPoint that will kind of go through some of the models we were looked at in lab, and then fair game also would be you being able to identify, some of the blood vessels that come off the aortic arch. You can see them here, the right subclavian, the right common carotid, the left common carotid, the left subclavian. So I'll make a little study tool for that. You may see, figures on in addition to the models I show you, you may see figures on a test which ask you to identify in yellow that you can see here some of the major artery branches. Okay. And again, I'm going to make a separate video for this. Some of the major venous branches are in yellow, which you would need to remember. Branches of the head. You can see there's only a couple here, although there will be a model that I show you that will show more so what's happening in the inferior view of the head and neck. Just a few veins for the head and neck, again in yellow, that you'd be responsible for. Abdominal, in yellow, that you would be responsible for, okay? Veins in yellow that you'd be responsible for. Celiac trunk branches in yellow that you'd be responsible for. There will be one part on a model I show you that shows the hepatic veins, but you're not going to need to be responsible for all the hepatic portal system veins. I'm just showing you what it looks like here. Some abdominal organs of the pelvis, again in yellow. Arms, this is a little closer look at some of the arm arteries that sometimes it's hard to see in this picture, that you need to be familiar with in yellow. Veins in yellow. Limb arteries in yellow. Veins in yellow. Okay. Now, I'm going to make a separate video that just uses mostly models and refers back to some of these for you to kind of focus on separately, because this video is getting a little longer than normal, and that will be related to the blood vessel lab. Okay. So we have gone through all of the items that we talked about in our objectives today. This is chapter 20, and have a good afternoon.